Never trust an antibiotic just because it works. This month, science proves some of them have been lying the whole time. January didn't bring loud biotech headlines. It brought quiet upgrades that might change medicine forever. This month, scientists figured out how to tell if antibiotics actually kill bacteria or just put them to sleep. We finally saw what DNA is really doing inside your cells. AI learned how to shut CRISPR off before it causes damage. And regulators, yeah, they're struggling to keep up. By the end of this video, you'll understand why January wasn't flashy, but why it might be one of the most important biotech months of the year. Let's start with something that sounds obvious, but really isn't. When doctors give you antibiotics, you assume those drugs kill bacteria, like dead, funeral, game over. Turns out, not always. A lot of antibiotics don't actually kill bacteria, they just stop them from growing. The bacteria basically hit pause. They're not multiplying, so lab tests say, yep, this drug works. But inside the human body, those bacteria can just wait it out. Once the drug is gone, they wake up and boom, infection is back. This is especially bad for diseases like tuberculosis and serious lung infections, where treatment already takes months. One wrong antibiotic choice and you're not curing the disease, you're just delaying it. That's the problem researchers at the University of Basel tackled this January. Instead of asking, does this drug slow bacteria down? They asked a better question, does this drug actually kill them? Their solution was kind of brilliant and kind of terrifying. They built a test that tracks bacteria one by one, single cells, under a microscope, over days, basically filming bacteria to see if they survive treatment or not. And when they tested this on tuberculosis bacteria and other lung infections, the results were shocking. Drugs that looked amazing in standard tests barely killed anything. Others that looked average turned out to be absolute bacterial assassins. This matters because antibiotic resistance isn't just about bacteria mutating. Sometimes it's about us choosing the wrong weapon. With this new test, doctors could one day match patients with antibiotics that actually finish the job. Drug companies could stop pushing meds that look good on paper but fail in real life. And maybe, just maybe, fewer infections come back stronger than before. So yeah, January exposed one of medicine's biggest blind spots, and that was just the beginning. For decades, we were taught that DNA is a code, letters, instructions, like a recipe book. That's not wrong, but it's incomplete. This month, scientists at Northwestern University, working with the 4D Nucleome Project, revealed something wild. DNA doesn't just sit there. It folds, it loops, it moves, constantly. Think of DNA less like a string and more like a tangled pair of headphones in your pocket. The shape matters a lot. Researchers created the most detailed maps ever of how human DNA folds inside real living cells. Not just one snapshot, but how it changes over time, from cell to cell. Here's why this is huge. Many disease-linked mutations don't sit inside genes. They sit outside, in so-called junk DNA. For years, that made no sense. How can a mutation outside a gene cause cancer or inherited disease? Now we know. If DNA folds differently, genes can be turned on or off at the wrong time. A mutation doesn't have to change the gene itself. It just has to change how the DNA is shaped around it. The team mapped over 140,000 DNA loops per cell type. They built full 3D models of entire genomes. They even created computer tools that can predict how DNA will fold just by reading the sequence. That means in the future, doctors might not just look at what your DNA says, but how it's folded. This changes how we think about genetic risk. It changes how we understand cancer, and it opens the door to treatments that fix DNA structure, not just DNA code. So January didn't rewrite genetics. It revealed the hidden layer we were missing. CRISPR is powerful. You already know that. It can cut DNA, edit genes, treat disease. But here's the uncomfortable truth. CRISPR doesn't always know when to stop. After CRISPR cuts its target, the enzyme can stay active. That leftover activity can damage healthy DNA or RNA nearby. It's like using scissors that keep snapping after you're done cutting. That's risky, especially in humans. Nature already has its solutions. Anti-CRISPR proteins. These are natural off switches that shut CRISPR down. The problem? They're insanely hard to find. In 10 years, scientists only discovered about 100. So this January, researchers decided to cheat with AI. A team from Monash University and the University of Melbourne used AI protein design tools to create brand new anti-CRISPR proteins from scratch, not copied from nature, designed. They focused on Cas13, a CRISPR design system that targets RNA instead of DNA. Using AI models, they designed proteins that could bind Cas13 and shut it off. Then they tested them in bacteria and human cells. The craziest part, the whole process took about 8 weeks. Normally, discovering a single anti-CRISPR could take years. This is a big deal because CRISPR therapies don't just need precision, they need control. On switches are useless without reliable off switches. AI didn't just speed this up, it changed what's possible. Another CRISPR breakthrough this month tackled a different problem, collateral damage. Some researchers noticed that Cas13 sometimes starts cutting DNA it's not supposed to. That's obviously bad if you're trying to treat disease, but a team publishing in Nature Biotechnology noticed something interesting. Cas13 only causes chaos when it's overused. High expression levels means messy cuts. Low expression levels means clean, precise targeting. By carefully controlling how much Cas13 was present in cells, they achieved strong knockdown of target RNA with almost no side effects. No widespread RNA destruction, no cell death. The takeaway is simple but powerful. CRISPR isn't dangerous by default. We were just being too aggressive with it. Between AI-designed off switches and better dosage control, CRISPR is starting to grow up. Now, let's talk about something that doesn't sound exciting, but absolutely is. Antibody oligonucleotide conjugates, or AOCs. Here's the simple version. Oligonucleotides are short pieces of DNA or RNA that can control gene expression. They're powerful, but they suck at getting to the right place in the body. Antibodies, on the other hand, are amazing at targeting specific cells. So scientists thought, why not glue them together? That's exactly what's happening. And this January, Nature Biotechnology highlighted AOCs as one of the most important drug types to watch. The biggest example right now is Duchenne muscular dystrophy. Companies are using antibodies as delivery vehicles to carry gene-modifying oligos directly into muscle cells. Early results show way higher protein restoration than older treatments. This isn't just better medicine, it's smarter medicine. Instead of flooding the body with drugs and hoping some reach a target, these therapies go straight to the source. And this approach could work better for brain diseases, heart diseases, and beyond. It's not a miracle cure, but it's a massive upgrade. AI is everywhere in biotech right now, but here's the uncomfortable truth. Most AI models are bad at real science. They sound confident, they use big words, but then they mess up basic chemistry. That's why In Silico Medicine launched something called Science MMAI Gym this January. Think of it like this. General AI models are like people who watched fitness videos once and now call themselves trainers. The gym puts them through real workouts. Models are trained on medicinal chemistry, biology, drug toxicity, and clinical reasoning. They're tested, measured, corrected, over and over, and the results are wild. One model improved its performance on drug discovery tasks by 10 times. 10. That's not a small upgrade. That's the difference between kind of helpful and actually usable. This matters because drug discovery is slow, expensive, and full of failure. AI that truly understands biology could cut years off development timelines, but only if it's trained properly. January showed us that AI isn't replacing scientists, it's finally learning to speak their language. All this innovation sounds great, but here's the reality check. The FDA is struggling. This January, reports showed leadership turnover, major staff cuts, and growing pressure on the agency. Over 20% of its workforce has been reduced. At the same time, it's being asked to move faster, approve more drugs, and even consider pricing issues. Despite this, the FDA still approved 55 new drugs and vaccines in 2025. That's impressive, but many were follow-ups, not brand new breakthroughs. The system is under stress. If biotech is accelerating, and it is, regulation has to keep up. Otherwise, life-saving tech gets stuck in limbo. January made one thing clear. Science is sprinting. Policy is jogging. Here's the big challenge tying all of this together. We're getting better tools, smarter tests, safer gene editing, AI that actually works. But complexity is exploding. Bacteria isn't simple. DNA isn't linear. CRISPR isn't binary. AI isn't magic. And regulation isn't built for speed. The risk isn't that biotech fails. The risk is that it succeeds faster. If you want monthly recaps of the recent biotech news, then subscribe for more.